Okay. Everyone able to see my screen?
Recording in progress.
Yes, Sudarshan.
So last class we discussed about the definitions, right? So what is study design, crossover, parallel study designs? Correct. What is site? What is multi-center study? What is multinational study? What is blindings? Still, we have to discuss few more definitions as well. Okay.
So, Sudarshan Swami here. Can you highlight the last point? The sponsor have recorded triple binding, right? Does that mean that the data, whatever sponsor will get, last point?
Okay. Generally, in triple blinding, all the three people we are hiding, right? Patients, investigator, and principal investigator.
Right. And
So
below.
we will be hidden means who have a record.
Mhm.
So sponsor have a record. The sponsor only will provide all this information, right?
Correct.
So sponsor will be having every time. Not only in triple branding, in every single, double, and triple also. Yeah, very good.
Okay.
Okay. See the next one, very important thing is the flow. Okay. So the flow will start. Okay, this is from where this was started. The stages you can say, or you can say flow also. First thing, first subject has to sign the ICF. ICF is nothing but informed consent form. Tell me what is informed consent form?
Agree. I mean, like the subject has to agree for treatment.
It's a legal document. Correct? It's a legal document signed by the subject. Okay. So that is called ICF, informed consent form. So this is the first form, subject has to sign. So what is required for us here? Mainly the date. Date we will capture in a data set level as well. The date is important for us when he signed the informed consent. So that is the first stage. First he has to sign the informed consent. Once he signed the informed consent form only, then he will enter to the next stage. That is screen. Screen. Okay. So in a screening, what we will do? We will screen the subject. Before giving the drug, what we'll do? We'll screen the subject. How will screen the subject? Tell me how will screen the subject? 100 subjects will come. All 100 subjects we won't take, right? Let us assume we took 60 subjects only. So remaining 40 subjects, how we eliminated? How we screen them?
Based on eligibility criteria?
Yes, based on the eligibility. In every protocol, if you observe, there should be inclusion and exclusion criteria, right? Eligibility criteria. Eligibility criteria. So based on the inclusion criteria and exclusion criteria, the subjects will be screened. Okay. So before giving the treatment, subjects will be screened based on inclusion and exclusion criteria. Okay, so now we screen the subjects also. Once the screening was done, the next important stage is randomization. Randomization stage means what we will do in this stage. Generally, this randomization will do the biostatistician will do this randomization. The randomization is nothing but so they will allocate the treatment. See which subject has to take the which treatment. So for example, here itself in this study, let us assume the parallel study. Who asked to take the 101 subject to drug A? Who told them? Who told them to 102 subject to take the placebo?
Investigators.
Even if how the investigator also knows. Even for example, 101 subject is taking drug A, drug B, placebo in this order. Why is taking in this order only? Why not is taking drug B, drug A, and placebo order? Who allocated this order? Who allocated this drug has to take this subject? So that will be decided in the randomization stage. Okay. So the drug allocation was done in the randomization stage. So this allocation also, it was not done in a proper particular pattern way. Okay, it will be done randomly. So they're going to allocate the drugs, they're going to allocate the treatment to the subject through randomization method. Okay. So first thing, biostatisticians will do this randomization. Okay, what they will do first, they will generate a code. They will generate the code. Okay, that is randomization code. Okay, so based on this randomization code, subject will be allowed to take the treatment. Means they're going to allocate randomly, they'll generate a code. Okay, each code represents, okay, this code, this code will come to the subject means, so in that code pattern, okay, in this pattern you have to take drug A, drug B, drug C, and drug C, drug A, drug B. So as per the allocation, the subject has to be treated as per the randomization code, as per the randomization sheet. Okay, so why they are going for a randomization method only? Why they are not going in another method? Why? So there's a specific reason you could definitely will be there. So understand what is the meaning of randomization. So important in the stage actually. If they're not followed a randomization method, let us assume there are 100 subjects are there in your study. 50 males, 50 females are there. Okay, so started the clinical trials, you started giving the treatment to the subjects. Treatment means the drug. The drug you give to the subjects. You completed your clinical trial process. Completed. That is, completed clinical trial process. You gave the drug. Maybe intentionally or not intentionally, you gave the drug only to the 50 males. You missed to give the drug for the 50 females. That is also drug A and placebo is there. Okay. Drug A will contain the active ingredient. Placebo will be the inactive ingredient generally. It's a sugar free, nothing will be there in placebo. So you gave a drug A to only 50 males intentionally or don't know. You missed to give the drug A for the females itself. You missed, you gave only placebo for them. You will get a reports, correct? You will get a report, the results also good. And you can send to the FDA also, no problem. But these reports are accurate now.
No.
Even though you got a report, those reports are not accurate. Why? You missed to give the drug for females itself. You don't know, maybe the drug may not work on a females. Correct? You have chances. And similarly, let us assume around same 100 subjects are there. 50 patients are there. 50 healthy people are there. You gave a drug, again, same. Intentionally, or I don't know. You gave drug only to the 50 patients. You missed to give the drug to the 50 healthy people. Same. When you completion of clinical trials, you will get a reports. Again, you can send those reports to FDA. But again, those reports are also not accurate. Because you missed to give the subjects, healthy subjects, you did not, for healthy subjects, for healthy patients, you did not give the drug also. So tomorrow, if they release the drug into the market, when some people who doesn't have any issue, if anyone took the drug, it may cause a problem. The chances are there. So like this, you're not supposed to miss anything. Any pattern you're not supposed to miss, either males or female patients or healthy people, or if you have a multiple drugs also, drug A, drug B, drug C. Sometimes you miss, you gave only in the order ABC, you forget to give an order A, BAC or CAB. Okay, so you have to follow everything. So if you follow any particular pattern, definitely you will miss something. To overcome this issue, they follow in the randomization method. So they're going to randomly, they will allocate to the subjects. So different patterns. So this will minimize your lot of your risk. So it minimize the completely. So the avoiding situations will be there, right? So you're going to cover all the criteria, all the situations when you follow the randomization. So biostatisticians will do this randomization, right? So investigators, based on this randomization sheet, the subject will be treated. Clear? So that is randomization stage. Once the randomization was done, so the next important will be treatment stage. Okay. So in the treatment stage, the subject will be treated as per randomization sheet. Randomization sheet or code. Yes. Subject will be treated. As once the treatment was done, treatment was completed, then we'll do the follow-up also. Once the treatment is completed, subject will be followed for some weeks or days. It means once the treatment was also given, completion also, will follow, means how is the health conditions, how it remaining will follow for some days about his health checkups and all. Okay, once the follow-up is done, yes, with this we're going to complete the study. Okay, so this is the flow you have to remember. First, always subject has to sign the informed consent. Next, it should be screening. Next, randomization. Next, subject will be treated. Next should be followed. Clear? This flow you have to remember. If you remember this flow, a lot of things are easy for you when you started working on a document. Because going forward, everything we are going to deal with the dates only. Okay. So if you remember this flow, what will happen is, for example, you got a 2 dates, one screening date, one treatment date. 2 dates, if you see the date, one subject have 2 dates, screening date and treatment. You derive the screening date and treatment date. Programmatically you derived or from the raw data you captured. By seeing the dates, what we have to understand, always your screening date is always before the treatment date only. Always your screening date is before the treatment date. Or if you have some randomization date and treatment date. So treatment date will come after randomization date only. And you have one treatment date and follow-up date. Always your follow-up date will come after treatment date only. Clear? So if you want to see the dates, okay, dates in which pattern it is coming is based on this flow. So always your informed consent signed date is before screening date. So to verify yourself, okay, once you check the data, you derived the whatever you derived the dates was correct dates or not. You can cross check with this pattern. That's the reason. Remember this flow. If you remember this flow, your lot of works was simple. Okay. See here in the study also. See, first screening, next to randomization. See, next to treatment, double-blinded treatment, end of treatment. Then you'll do the follow-up. Finally, end of study. Study going to be completed. Okay. Screening, randomization, treatment, follow. Clear? Here, each stage is there, right? Screening, randomization, treatment, follow-up. Majorly these 4 only. Each stage, each phase is one element. There's one more derivation, definition, element. Element, it is nothing but each stage or each phase is one element. It's nothing but each stage, each phase. Each, you can say each clinical trial phase. Each clinical trial phase is one element. So screening one element, randomization one element, treatment one element, follow-up one element. What are the elements you have? Screening, randomization, treatment, and follow-up. Okay, these are the 4 elements you are going to have in every study. It should be same. These are the 4 standard elements. Next, epoch. What is epoch? A group of elements. A group of elements will called as an epoch. Will called as an epoch. Means all these elements together we called as an epoch. Okay, clear? All these elements together we're going to call as an epoch. Okay, going forward in domains while we're discussing, so these things will come, elements and epochs. Okay. So if it is screening epoch, if it is randomization epoch, treatment, particular treatment epoch, and follow-up epoch. Okay. So this all the elements together we call as an epoch. So sometimes this treatment stage also, in this treatment stage, he may not take only one treatment. Let us assume he's taking drug A and drug B. Two drugs is taking. So it's going to be your screening epoch, a randomization epoch. When it comes to treatment epoch, either in the treatment, which treatment is a drug A epoch or drug B epoch? Okay, or else first treatment epoch or second treatment epoch? First treatment epoch or second treatment epoch? And follow up. Okay, so all the together we called as an epoch. Each stage is one element. Okay, so these two things also you have to remember. Not only. Okay, and apart from this, if you observe this study design, you'll be having see visit 1, visit 2, visit 3, visit 4, visit 5, visit 6, 7, 8, 9. So these visits are there in the different visits. The screening day 0, week 2, week 4, week 8. What is this all are? These are scheduled visits. So there are two types of visits will be there. Scheduled visits, unscheduled visits. Scheduled visits and unscheduled visits. Scheduled visits are nothing but planned visits as per protocol. So in each protocol, definitely they're going to mention some planned visits. So when a subject has to visit the site, it was already mentioned in the protocol. So these are planned visits. Unplanned means, for example, visit 4 is in week 4, visit 5 is in week 8. So between week 4 and week 8, in between he came to the hospital again. Maybe due to anything, due to some adverse event or some problem to check something, he visited the hospital, he visited the site. Then it's going to be unscheduled visit. Correct? Unplanned visit. So unscheduled visits is nothing but unplanned visits. Okay. So scheduled visit, the visit, planned visits, screening visit. Okay. Or it's screening. You can say screening or randomization. Or next is week 2, week 4. So it is day 0 they are mentioning. Day 10 means 10 days before the randomization will be the screening visit. First visit. Okay. Next day 0. Or week 1. Week 2. Week 4. Week 8. This is going to be scheduled. Unplanned means unscheduled visit. Unscheduled visit 1, visit 2, visit 3. Unscheduled visit 2. Okay, you may have between 2 scheduled visits or planned visits, you may have one unscheduled visit or multiple unscheduled visits also was there. So that's also possibility there. Okay, basically unscheduled is nothing but unplanned visit. Okay, like this visits also there are 2 types of visits will be there. Okay, that's it. the definitions. Any questions, anyone?
Sudarshan, I just have one question. So
Yeah.
on the other side of the book, right, I could see like a baseline and randomization, right? So both are same or like any difference in between baseline and versus randomization?
No, baseline versus randomization. It's they are mentioned. Okay, they gave the randomization as a was baseline value.
Mhm.
Okay, this is also one visit. They are naming the randomization as a baseline.
Mhm.
Yes.
So both are same, right?
Both
Just
are
to,
same.
it's difference in wordings. That's it.
Yes, that's it.
Okay.
So basically, we will use a word called randomization only in all the study.
Mhm.
In this protocol, they are mentioned as a base value or baseline value. That's it.
Mhm.
They gave
Okay.
naming as a baseline. Okay.
Mhm.
In another study, they may not mention as a baseline. They kept as a
Mhm.
randomization also. Okay.
Okay.
Randomization default word. Everyone uses.
Mhm.
Okay.
Okay. Got it.
So
Thank
I
you.
gave you notes. So these are the standard elements. Screening, randomization, treatment, follow. Okay. Any questions, anyone? In the
And
definition.
also epoch you mentioned is a group of elements, right?
Correct.
So here let's say randomization is also one epoch. That's it, right? It considered as an element, and
Yes,
we
it's
can call it as
considered an
epoch too.
element. So, see, each one is one element.
Yeah.
Randomization is one element, treatment one element. All together we call it an epoch.
Mm-hmm.
Okay. The
Mhm.
definition is called together we call an epoch. But
Mhm.
when you go for a derivation level, it was you'll understand there you will know clearly.
Okay.
Generally, epoch is nothing but particular subject. If any date will comes under, it
Mhm.
falls under which stage? It falls under treatment stage or randomization stage or screening stage.
Mhm.
They will derive epochs. Okay.
Okay. Mhm.
Elements will derive separately.
Mhm.
Okay. So just
Okay.
understand the meaning. Okay. Tell
Mhm.
okay, these are the elements all together we call epoch.
Okay.
As of now, then we understand when you are doing the programmatically, you are more you get a clarity.
Sure.
Is there any abbreviation for EPUB?
No, there is no abbreviations. It's epoch only. Okay. Fine. So 1 second before we start one topic, just give me 1 minute. Continue. So first we'll start with an SDTM. In SDTM, first we'll discuss about the workflow. Okay. So this is an important thing. If you go for an interview, the first thing they will ask your work. How is your day will start, how your day will end. How is your work will start and how your work will be ended. Okay. Completely flow I'll explain. Okay. See the first one. First, they are going to assign a one study. Study is nothing but a project. Anything, one project or study they will assign. Once they assign the study, what they'll do, first they are going to give some documents to you. You have to review those documents. What are those documents? Already I shown you, right? One protocol document they will give. Okay. One plain CRF, the CRF document. One protocol document like this one protocol document and a one empty CRF document they'll give. So we already discussed it, right? What is CRF and all? So like this one CRF copy they'll give. Okay. Next. Yes, they'll allocate the project means it will be having the raw data sets. Okay. It should be having the raw data sets. For example, if I send a project, the project will be having the raw data sets like this. You can also go through those raw data sets. Next. On SAP document, there is statistical analysis plan. In SDTM level, it won't useful much, but in ADAM level or in a tables level, this SAP will be useful. It contains how to derive some variables. Okay. Some variables, how to derive some variables. There's an imputation concept will be there, imputation concept. Some mock shells should be there. It will contain, the SAP contains everything. HDTM level at not required. This is an additional document. Along with the study, they're going to share. So you have to go through these documents first. So they'll give you sufficient time to go through it. Then later in the study, the next step is ACRF. ACRF is nothing but annotated CRF. So when any new study, when we start any new study, at the starting it will be in empty only. The CRF should be empty like this. So they'll give an empty CRF study. They'll ask you to annotate it. I'll show you how your annotation CRF should be. So this is your annotation CRF. It looks like this. So annotation CRF is nothing but in a CRF, each information has to go to which domain and which variable that we have to annotate it like this. For example, in the CRF, demographic information is a date of birth information. Okay, date of birth information has to go to which domain? It has to go to DM domain. In DM domain, there is a variable called BRTSDTC. It has to go to this variable. For example, gender information is there. Gender information has to go to which domain, which variable. So in a DM domain, sex variable. So going forward, we're going to discuss what are the domains we have in SDTM level. In each domain, what are the variables we have also we're going to discuss. And each variable, what is the meaning of that variable. So according to that meaningful variable, you have to assign, you have to annotate it here. Okay, generally this annotation thing will be taken care by your senior statistical programmer or a principal statistical programmer will take care. Now able to see my screen? Everyone confirm? Hello?
Sudarshan, your voice is very low.
Yeah, it is clear. We can see that.
Okay. Able to hear me clearly?
Yes, sir.
It is clear, sir.
Okay, fine. So first this annotation, this is what annotated CRF. So in annotated CRF, every information in the CRF should be annotated like this. Means that information has to go to which domain and which variable. Okay, so this CNET annotation was also done with some set of rules and regulations. Okay, let me give some, there are simple rules only. Some guidelines will be there. Okay, CDISC gave some guidelines. So these are the CDISC guidelines. Okay, I'll share this document to you guys as well. Gone through it. Very simple guidelines only. So means how to annotate it. Means where to place this name. Okay, if information is available, where to place this information. Okay, where to place this variable name. Especially here. Okay, what's the font you have to follow? What's the font size, page orientation, page size? You have to bookmark that one. Okay, so font colors. This is you have to follow 12 point times Roman if possible. Guys, this workflow we'll discuss tomorrow. I don't know something. This happened. Just continue disconnect. Okay.
Yeah, sure, Sudarshan.
I don't regret.